Hello, everyone, and welcome to today's live broadcast. Combine immunohistochemistry and RNA in situ hybridization to elucidate tumor molecular heterogeneity in prostate cancer. Presented by Nallasivam Palanisamy. He is an Associate Senior Scientist at the Henry Ford Health System. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Advanced Cell Diagnostics. For more information about our sponsor, visit acdbio.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Nallasivam Palanisamy. I will now turn the presentation over to him.
Hello everyone, my name is Nallasivam Palanisamy. Today I'll be talking about combined immunohistochemistry and RNA in situ hybridization to study the tumor molecular heterogeneity in prostate cancer. The outline of my talk is going to be first on the comparison of RNA and the protein expression by immunohistochemistry and RNA-ISH. So this is strictly for validating how comparable RNA in situ hybridization is to immunohistochemistry. Then I will show you how we can detect the ETS gene fusions in prostate cancer. As many of you know, there are many ETS-related gene fusions have been identified in prostate cancer. Since we don't have good antibody for all of the ETS gene fusions, we have developed the RNA in situ hybridization procedure for the tissue-level expression of these genes. And then by using this method, we show that how we can combine both immunohistochemistry and RNA in situ hybridization for the detection of more than one markers in a single hybridization experiment. So with this approach, we will be able to study the tumor molecular heterogeneity in prostate cancer. As you may be aware, more than 80% of the prostate cancer are multifocal disease. So we are trying to understand the genetic underpinnings in the different tumor foci by combining this RNAs and immunohistochemistry in a multiplex approach. And there are several non-coding RNA targets have also been identified to be specifically expressed in prostate cancer. Since we cannot develop a good antibody for non-coding RNAs, so we developed RNA in situ hybridization probes for the reliable detection of the non-coding RNA at the tissue levels. And last, I would like to show you how we can combine both DNA fish and RNA-ISH if you are interested in actually for the detection of two molecular markers. So all the data that I'm going to present in this presentation is about the prostate cancer research that is being conducted in my laboratory. And the both RNA-ISH and the immunohistochemistry procedures that I'm going to show is completely under the bright field microscope application. So you can go up to like 4 markers that can be detected under the bright field without the need to use the fluorescence. Okay. So here, as my laboratory is one of the early adopters of the technology of RNA in situ hybridization back in 2010, about 6 or 7 years ago. So I thought it would be useful to share some of my experience as useful tips for some of you who may be just beginning to set up this RNA in situ hybridization in your laboratory, or who some of you may be interested in start using this technology. I thought this would be useful. So I would recommend to use freshly cut FFPE tissue sections when you are doing RNA in situ hybridization. Slides that are cut within about 3 months are ideal. If they are more than 3 months old, I would recommend actually test a small batch of slides using a positive and a negative control probes to assess the RNA integrity. And you can perform the RNA in situ hybridization both by manual methods and automated methods. So in our laboratory, we have actually used the Ventana Discovery XT, the research instrument for performing RNA in situ hybridization. And then the other important thing that I would like to mention is if you are planning to do both IHC and RNA-ish to detect more than one target in the tissue of your interest, I would recommend actually testing both your antibody and the individual RNA-ish probe first individually, make sure they are performing well before combining them. Then other important point is make sure that if you are doing both IHC and RNA-ish, make sure you do the RNA-ish first, then do the IHC, not the other way around. And as I mentioned before, the DNA fish and the RNA fish can be performed on the same tissue as long as your targets are distinct. And other important thing is, it is important to know, have some clear knowledge about the location of the target markers. So especially when you're doing a multiplex assay like the antibodies, make sure that you're expecting the staining in the nuclear or cytoplasmic or membranous, like that. If you have a clear knowledge, it will be easy to interpret your results. So these are some of the important tips that I thought it would be useful for beginners. And if you have any more questions, please refer to the link that I have provided at the end of this slide, where you will find many useful tips that is more related to the RNA-ish protocol. So now I'm going to talk about the prostate cancer molecular markers, how you can actually detect these molecular markers at the tissue level by combining both immunohistochemistry and RNA in situ hybridization. As you can see, there are several markers that have been identified in prostate cancer, particularly the ERT gene fusions like the ERG, ETV1, ETV4, and ETV5. These are the ERT family gene rearrangements that are detected at varying frequency. And these markers are expressed only in prostate cancer, and sometimes you will see in high-grade and low-grade cancer as well. And these markers, it is important to note that they express only when there is a chromosome translocations. The expression of these genes are activated by chromosome translocations. Otherwise, you don't see any even basal level of expression of this protein. So that makes it ideal for doing this type of experiments. And similarly, we found another target is PINK1, which is identified in about 10% of the prostate cancer. Then the ETV, the PCA3, PCAT1, SLAP1, and KLKP1, these are the non-coding RNA targets that are well characterized in prostate cancer. They are associated with some prostate cancer development and its outcome. So since we have this kind of variety of these markers, so we are interested in developing methods that can be routinely used for the reliable detection of these markers at the tissue level. Okay. Since we have these many markers and encoding RNA, so we cannot have or we cannot develop a method that can be performed by a single approach. Say, for example, we cannot do just by immunohistochemistry. So we have to develop both RNA hybridization and immunohistochemistry depending on the availability of the antibodies. So here for ERG and SPINK and P10, we have good antibodies. So this can be used for immunohistochemistry. So all other markers, we don't have good antibodies, and they can be detected only by RNA hybridization. So since we have antibody for ERG, P10, and SPINK1, so we can actually do the dual color or the three-color immunohistochemistry, like combining ERG, P10 or ERG, SPINK1 or the SPINK1, P10, like that. You can do this combination. So we have published this work. And for ETV1, ETV4, and ETV5, we have to perform on the RNA hybridization. Then similarly, for all the non-coding RNA targets, we have to use only RNA hybridization. So having developed both IHC and RNA-IHC, so we were interested in studying in these markers in a multiplex mode rather than studying each marker individually, which will be time-consuming, laborious, and sometimes you are limited with the availability of the tissue specimens. So for those reasons, we are trying to develop a multiplex method where we can detect more than one marker in a single experiment. So we have optimized the protocol, which is consistently working, so we can detect up to 4 markers in a single experiment by combining like 2 markers for IHC and 2 markers for RNA-IHC. So we will be able to do both dual IHC and dual RNA-IHC, so that you will be able to detect up to like 4 markers in a single hybridizations, which I will show you in the latter part of this presentation. So before going into the details, so when we started using the RNA hybridization, so we did our own due diligence experiments to see how RNA hybridization is comparable to immunohistochemistry. So for that, since, as I mentioned before, since we have a good antibody for ERG gene, so we got both antibody and we developed the RNA hybridization probe for ERG. And then we were trying to compare the performance of the RNA versus immunohistochemistry. So the next slide, as you can see here, I'm showing a tissue sections where we hybridized with the ERG antibody, where you will see the specific staining of ERG only in the tumor area, and there is no staining in the stroma or in the benign area. So you will see such a specific staining with the immunohistochemistry. But when you do the RNA hybridization, so you will be able to see similar results where, as you can see, the RNA signals can be seen as distinct punctate dots, and you will see most of these signal are in the cytoplasm. As comparable to the immunohistochemistry, you will see the specific staining of the RNA molecules only in the tumor area, nothing in the stroma or in the benign areas. Okay. So based on this, we are quite convinced that, we can perform both RNA hybridization and immunohistochemistry, which is quite comparable. So if you don't have a good antibody for any gene of your interest, I think RNA-IHC may be a viable option to consider for doing the tissue level expression of any gene of your interest. Okay? So then, based on the level of expression of your target, you will be seeing a different level of RNA staining. So you will see when it is negative, you don't see any dots. Suppose if the protein so the RNA level expression is kind of low, you will be able to see only like few dots. Then if the level is moderate, you will see like multiple dots. If it is higher, you will see more like diffused. You will see more like a diffused pattern of signal. Okay, so we developed, based on the level of expressions, we developed this RNA hybridization scoring criteria, which you can do it manually by 0 to like 1+, 2+, or 3+, and 4+. So this will help you to score the RNAs. But I think recently there are methods have been developed for digital quantitation of the RNA hybridization, where if you have a slide and get it scanned with a high resolution scanning or a digital imaging, that will help using digital image processing software, where you will be able to get a quantitation of the RNA signal as well. So either you can do it manually or by the digital methods. Okay? So having identified the several markers in prostate cancer, as you can see, like S gene fusions, SPINK1, and the RAF kinase gene fusions, this is the first time a molecular classification of prostate cancer have been developed. And it is not only that individual markers, they are identified in a distinct subset of prostate cancer. Recently, there are many preclinical studies have been completed showing the potential that some therapeutic drugs targeting these S gene fusions, SPINK1 or the RAF1, have been developed, although these drugs are not ready for the prime time clinical use, and there are active research going on to bring it to the clinic targeting this. So given the identification of several molecular markers in prostate cancer, identifying distinct molecular subsets, and with the development of therapeutic inhibitors targeting these molecules, I think it is very important that we need to have a method that is much reliable, and it is very robust for the reliable detection of these molecular markers in a given tissue, whether it is a biopsy tissue from the metastatic prostate cancer, or from the initial diagnostic biopsy, or from radical prostatectomy specimens. We should be able to classify these patients based on these molecular markers. So when the drugs are available for clinical use, and I think it will be very useful for identifying the patients carrying the specific markers, so that they can benefit by selecting the appropriate therapy. So with that idea in mind, we want to develop a much more reliable kind of detection method for the detection of these molecular markers. So here I'm showing you a scheme, a flow diagram, if you want to screen the prostate cancer markers. As you can see, if you get a tissue biopsy, needle biopsy or prostatectomy samples, it goes to the pathological examinations. They do the morphological analysis, whether there is a cancer or it is normal. So when it is a cancer, like if there is a very small kind of atypical lesions, there may be additional staining can be done to rule out whether they are cancer or benign, like doing the high molecular weight cytokeratin and P63 and AMA-CS staining to rule out the small atypical glands. So once any tissue specimen, if it is confirmed with a cancer, so if you want to go for screening for molecular markers, so like you have the ERG, SPINK1, ETV1, ETV4, ETV5, and BRAF, all these markers. So if you have to screen these markers individually, you have to do at least 5 or 6 different slides. So if you do it, sometimes, because of the limited amount of the biopsy tissue, you may not have the luxury of having 5 or 6 slides. Okay, so in that situation, you should be able to screen more than one marker in a single hybridization experiment. So here, other important thing that I want to emphasize that these molecular markers, although we have several molecular markers, so far based on the research, what is available in the literature, these molecular markers expressed in a mutually exclusive manner, meaning that you don't find like more than one marker that is expressing in a given tumor foci. So you may see more than one marker in different tumor foci in prostate, because prostate being a multifocal cancer, but you don't see like more than one marker expressed in the same nucleus like that. So that gives another unique advantage of actually developing multiplex method. It is easy to resolve and interpret the results. So for that reason, like I mentioned, that we can develop both IHC and RNA-ish methods. Like for ERG and SPINK1, we have good antibody, we can do the IHC. Then for ETV1, ETV4, and ETV5, so far, it is fish fluorescent in situ hybridization using the break-apart fish is the ideal method. But now we have developed the RNA in situ hybridization for the tissue level expression of the S gene fusions. So in the next slide, I will show you So here I'm showing the example of doing both IHC and RNA on individual slides. So here I'm showing the ERG on the needle biopsy. So we did the immunohistochemistry where you will see the specific staining only in the tumor. Here I'm showing the SPINK1 and the needle biopsy showing specific expression only in the tumor, and both ERG and SPINK1 are done by IHC. Here I'm showing ETB1 expression by RNA in situ hybridization. As you can see, it is specific only in the cancer, but you don't see any non-specific staining in the benign. And similarly for ETB4, we developed the RNA in situ hybridization methods, which is actually staining specific only in the tumor, but not in the benign or stromal tissue. So which clearly shows that, we have individually tested these immunohistochemistry antibodies and the RNA in situ hybridization probes for the reliable detection of the ETS gene fusions in prostate cancer. So we have published this work a couple of years ago, so you will find more details about the probes that we used for the RNA in situ hybridization and the antibodies, and how reliably you can detect the gene fusions by doing both IHC and RNA-ISH in prostate cancer. So I would recommend go through this paper for more details. So next I would like to show an example how actually we will learn some new information or some unexpected results, when you develop some new approaches. So here I'm showing one example. So this is a prostatectomy samples where there is a tumor foci that is stained with a hematoxin eosin, and which we, before the RNA in situ hybridization, we confirmed this case was positive for ETB1 rearrangement by FISH. So when we developed the RNA in situ hybridization methods, we stained this with RNA-ISH, and we were able to see a good staining of RNA-ISH all over most of the tumor area, and except there are two areas, they are still cancer, where we did not find any staining for the ETB1. So next what we did out of curiosity, since these are all still cancer but they are negative for ETB1, so on a consecutive slide we did the ERG immunohistochemistry, and we found that the area that is negative for ETB1 was positive for ERG. So this is for the first time we were able to see that this is called intra-tumor heterogeneity, because at the histological level on the H&E, the pathologists, they call it, actually they are all the same Gleason grade, but not with much variation. But there are tumor areas, distinctly separate at the molecular level, where we were able to see both both ERG and ETB1 present within the same. And this is called intra-tumor heterogeneity, where we clearly demonstrated that the presence of ERG and ETB1 within the same tumor foci. But here, as I mentioned before, these markers are mutually exclusive. So we are not seeing both ETB1 and ERG in the same cell, but they are in two separate areas. So this emphasizes the need for not only the morphological classification, it is important to include the molecular classification of the prostate cancer. So now we are trying to understand what it means when you have a patient with more than one marker present in the tumors, whether it makes the cancer more aggressive, or whether you'll have early biochemical recurrence or metastasis like that. We are trying to correlate the outcome or the importance of this more than one marker positivity in prostate cancer. Okay? So next, the important thing is, when we see this kind of intra-tumor heterogeneity, so that makes us to think that more than about 87% of the prostate cancer are multifocal, 87% of the cases are multifocal cancer, only about 25%, only about 12% of the cases are like a unifocal disease. So when you have a prostate cancer with more than one tumor foci, so now we are asking the questions whether these individual tumor foci are they genetically identical or they different. Okay. So, and most of the time, the dominant or the index nodule, they call it as the largest nodule in a multifocal disease, is presumed to harbor the most aggressive biological behavior. and therefore dictate the overall clinical behavior of the prostate cancer. But it is not always the case that when you have the largest tumor volume or the highest Gleason grade or the extra prostate extension, you may not find everything within the single tumor nodule. Sometimes you will see more than one tumor foci may have the, you may see the largest tumor with a low Gleason grade, but another tumor foci may be with a higher Gleason grade, but the size may be smaller than the index nodule. Like that, so you may not see like everything within the single tumor foci. So therefore, more than about 1/5 of the cases, more than about 1/5 of the prostate cancer, you may not find the largest tumor volume or the highest Gleason grade or the stage. They are not present within the same tumor nodule in nearly about 1/5 of the cases. So sometimes you will find some smaller kind of tumor foci which with a Gleason nodule that is at least more than one Gleason grade higher than the so-called the dominant or the index nodule, like that. So it is for that reason, just by studying the so-called the dominant or the index nodule may not give all the complete information. So now I think it is there is a consensus that it is important to consider even the secondary nodule also to get an integrated analysis to make a collective decision like that. So it is for that reason there are many major kind of centers. They started adopting the whole mount prostate tissue analysis rather than looking at only the dominant nodule or the index nodule, like that. So when with that idea in mind, we wanted to study the tumor heterogeneity. We wanted to study the tumor heterogeneity in prostate cancer. So as you know, the heterogeneity in prostate cancer is the reason for the difficult diagnosis and sometimes prognosis of the tumors. When you have a multifocal tumor like this, you have not only one tumor foci, you have several tumor foci, it will be very difficult to understand what is going on in each tumor foci. So the clinical significance of smaller secondary tumors and the relationship between the extent of molecular changes and the metastatic potential of individual tumor is not clearly understood. Okay, as I showed before, you may be having like a different kind of clonal population of cells driven by the different driver molecular aberrations in these cases. So it is for that reason, we have to have, so it is not only on the hormone prostate cancer. So we have done a kind of a pilot study a few years ago where we showed that we can demonstrate this type of tumor heterogeneity even in the needle biopsy samples as well. As you can see here, when you have a needle biopsy, you have one tumor foci in one end of the tumor biopsy, then another tumor foci in this one. So what we call it as a discontinuous foci. Okay. When you have these kind of two different foci separated by a benign tissue, how do you interpret this? Are they coming from the same tumor, or they are coming from two different kind of tumor, and they are separated, kind of spatially separated by benign or stromal tissue? So under these circumstances, what is happening? Whether these two tumors are identical at the molecular level or they different. Okay, so for that reasons, when we did a study by using only ERG and SPINK1 by dual immunohistochemistry, we were able to demonstrate that about 13% of the needle biopsy had about one ERG foci positive and the SPINK1 foci positive. So the end, they are not the same. Okay, so you'll see one tumor is positive for ERG and the other tumor is positive for ERG. So out of the 97 needle biopsy cohorts from 80 patients, we found with the involvement of 20 to 100% cancer involvement, we found about 25% of the cases they had clonally distinct cancer foci, and they are discordant ERG and or SPINK1, like that. Okay. So it is not only, we don't have to look at the whole mount prostatectomy samples, even in the needle biopsy also, we can demonstrate this type of heterogeneity. So it is for that reasons, so I think combining more than one markers in a multiplex approach will reveal some important information which we were not aware of before. So here I am showing an example, an intertumor molecular heterogeneity detected in the needle biopsy sample. So here there is one tumor foci at this end of the biopsy. There's another tumor foci in this biopsy. So here we did combined both the IHC and RNA-ish, where we see this foci is positive for ERG and the other tumor is positive for ETB1. So this is a very clearly, as you can see, this is the zoom-in view of both ETB1 and ERG, where we can clearly demonstrate it. Two distinct foci, distinct for different molecular aberrations. One is ERG, the other is for ETB1, separated by a high-grade pin or a somal tissue. So this is the type of molecular heterogeneity that you can identify by combining both the IHC and the RNA in situ hybridization. Okay? So therefore, so like I discussed before, we have these multiple markers. Okay? So based on the availability of the reagents, and we were able to kind of improve the detection approach so that we will be able to detect at least 4 markers in a single hybridization right now. Okay, so we can combine both IHC and RNA-ish. So we have 2 markers for IHC. That is ERG and SPINK1. These 2 can be detected by IHC, and ETB1 and ETB4 by RNA-ish. So we will be able to detect all these 4 markers on the same tissue by doing a more sequential hybridization of RNA-ish first and followed by the immunohistochemistry. So this is a 3-day protocol, so when we do it manually. So first day you do the antigen retrieval and do the RNA in situ hybridization, and the second day you can detect the RNA-ish, and then do the primary IHC antibody. If you can leave it overnight, and the next day you can detect the, and the third day you can detect the IHC. So that you will be able to see all the 4 signals. So here it is important that always do the RNA-ish first, followed by the IHC. Okay, don't do the IHC first and the RNA-ish, so you will not be able to see the RNA-ish signal. Okay, here I'm showing an example where on a tissue microarray we are fortunate to have like the tumors that are positive for all 4 markers. Okay, so this is the tissue microarray where we hybridized both IHC and RNA-ish. Okay, so we did ERG, ETB1, ETB4, SPINK1, all the 4 markers, and we were able to detect simultaneously by doing a sequential hybridization of RNA-ish and IHC. Here I'm showing clearly there is a tumor sample that is positive for ETB4. As you can see, the strong staining, and this is the SPINK1. There is a strong blue staining, more heterogeneous. And this is the tumor that is positive for ERG here, and this is another tumor that is positive for ETB1. So for that, we will be able to detect all the 4 markers on a single slide. So this is, I think this approach is very useful, especially when you are limited with the amount of the tissue that is available for hybridization. So like if you have a biopsy tissue from bone mets or lymph node biopsy, or a needle biopsy, which has a very small amount of tumor, so you don't have the luxury of cutting multiple slides. So you may not be, so if you want to screen all the 4 markers, you don't want to waste 4 slides, you'll be able to see with the availability of a single tissue, you'll be able to get this kind of valuable information. Okay. Next, this is not only on the needle biopsy or on the TMA. Here I'm showing a whole mount prostate tissue where we were able to see there are 5 different tumor foci. This is a multifocal prostate cancer with the 5 different tumor foci. And initially, we screened this tumor with only ERG and SPINK1 dual IHC. With that, we were able to see the so-called the dominant tumor nodule, which was negative for both ERG and SPINK1. And this tumor foci was positive for ERG, and this is positive for SPINK1. this tumor foci is negative and this tumor foci was positive for ETB1, which was done on a consecutive slide. This area was positive for ETB1. So here we clearly demonstrated that out of the 5 individual tumor foci that are spatially separated by the benign or the stromal tissue, and they have a distinct driver molecular aberrations. As we know that these H gene fusions are the driver molecular aberrations in prostate cancer, and we see that the tumors of the different Gleason grades, so like the ERG foci is like a Gleason 6, and whereas the tumor foci that is positive for ETB1 is more like a Gleason 7. So and the so-called the dominant or the index nodule, which is again it has a Gleason 6 pattern. Okay, so given then further we have done actually P10 IHC on the same cases where we found the P10 was deleted only in the tumor foci that is positive for ETB1 with the high Gleason grade, whereas all other tumor foci were negative. The P10 was intact. So like that, we will be able to kind of develop or gather such a valuable information just by combining both immunohistochemistry and RNA. Okay, so now it is important. Okay, so one can ask the questions. And when you have this multifocal tumor with multiple driver aberrations, which tumor foci is kind of biologically important, which is going to actually contribute for the metastasis or the aggressive nature of the treatment like that, and also how these patients are going to respond to the standard treatment like that. Okay, so I think these are all some of the questions that are we are actually beginning to address by using this type of approach. Okay, so so here I want to emphasize the point that although the ERG and ETB1 belong to the same H gene family, but at the biological level they behave entirely different way. So the ERG and ETB1, they control a common transcriptional network but largely in an opposing fashion. Okay, so the ERG actually negatively regulates the androgen receptor transcription program, and ETB1 cooperates with AR signaling by favoring activation of AR transcriptional program, and ETB1 expression, but not that of ERG, promotes autonomous testosterone production. ETB1 but not ERG upregulates expression of AR target genes as well as genes involved in steroid biosynthesis, and ETB1 supports the development of invasive adenocarcinoma under the background of P10 loss. Okay, so the distinct biology of ETB1 associated with prostate cancer suggests that this disease class may require new therapeutics directed to underlying programs controlled by the ETB1. So this clearly emphasized the need okay, to classify the tumors based on the molecular aberrations, and although these H genes, the family, are involved in prostate cancer, they are expressed in a mutually exclusive manner in the two different tumor foci. So it is important, and they work entirely in a different molecular mechanisms. So the drugs targeting these molecules, we cannot use, although they belong to the same H family. You cannot actually treat all of them as the same. So we have to develop individual therapeutic molecular targets. So these are all some of the new information that we are learning out of this by doing this molecular heterogeneity analysis. And this is the new beginning where we can actually use this information to develop new treatment strategies. Okay, I think with that I have convincingly shown you the power of the combined immunohistochemistry and RNAs, how you can actually study this tumor heterogeneity in prostate cancer. As I mentioned, that this approach is not limited only to prostate cancer. So this approach can be adapted to any other solid cancer as long as you have these kind of distinct molecular markers and you know the localization of these markers and the expression pattern. And I think with this kind of approach, we don't have to have this sophisticated method or anything like that. We will be able to learn a lot of new information. So because this type of tumor molecular heterogeneity has been studied by different approaches like the next generation sequencing. So like people have done like micro dissection of individual tumor foci. They have done next gen sequencing. and they have demonstrated that there are multiple molecular aberrations, with the inserts and deletions, multiple gene fusions are occurring in different tumor foci. But those studies did not reveal any targetable molecular aberrations. So therefore, we decided to stick with the already established molecular markers, which are known to be driver molecular aberrations in prostate cancer. And the drugs are in the preclinical studies showing the potential of developing specific therapeutic drugs targeting each molecule. I think when these drugs reach the clinic, when they have the therapeutic potential, I think the approach that I described, the combined IH and RNA-ish, will be the powerful approach for the early diagnosis of the needle biopsy or the prostate metastatic biopsy for characterization of these tumors for the presence or absence of these molecular markers, which is a very powerful kind of approach. Okay. So next I would like to move on to show some of the non-coding RNA targets in prostate cancer. As we know that non-coding RNAs, there is a general notion that these non-coding RNAs are not coding for any protein, but which is not 100% true, because it has been shown that recently there are many non-coding RNAs. They do indeed code for some small proteins, what we call it as micropeptides, that has been shown recently by some of the work by Dr. Eric Olson. So the non-coding RNAs, you cannot strictly say that they are strictly non-coding. Some of them are making proteins, and eventually antibodies can be developed. But right now, due to the lack of information, so we generally use the RNA in situ hybridization approach to detect the non-coding RNA expression in tissues. Okay, because the function of the non-coding RNA is more kind of generalized mechanism. Like non-coding RNAs act through a variety of mechanisms such as remodeling of chromatin, transcriptional coactivation, protein inhibition, post-transcriptional modifications, or they can function as a decoy elements, or the more that function is more the regulation of other protein coding genes. So given the importance, because there are several prostate cancer specific non-coding RNAs have been identified, like PCAT, PCA3, and SLAP1, and KRSP1, like that. These are all the prostate cancer specific non-coding RNAs. So we have developed some methods to see the expression pattern of this non-coding RNA at the tissue level. So I think without the RNA in situ hybridization, so either we can do PCR or some other quantitative method to study the expressions. But if you want to see the spatial, just preserving the spatial morphology of the tumor, and you want to assess the expression pattern, I think RNA in situ hybridization is the best approach. So here I'm showing one example where we identified a new gene fusion involving a KRSP1. Here we developed an RNA in situ hybridization probe where we showed that very clear kind of tissue-specific expression of the RNA molecule only in the high Gleason grade, like 4+3 or 3+4 high Gleason grade prostate cancer, but they are completely negative in the benign prostate and also in the low Gleason grade prostate. So we found this kind of non-coding RNA that is expressed only in the high Gleason grade specific manner. Then next, the PCA3 is a very good non-coding RNA that is a prostate cancer specific. It is expressed almost about 90 or 95% of the prostate cancer, and which is can be used as a marker. And it's important is that this PCA3 RNA transcript can be detected in the urine. It is not only PCA3, the TMPRSS to ERT gene fusion can also be, fusion transcript can also be detected in the urine. So recently there was a test that has been developed and that was approved by the FDA for the PCA3 urine detection of the PCA3. At the University of Michigan, there was a new test that has been developed for patients who has elevated PSA, and those patients by non-invasive methods we can detect both the PCA3 and the TMPRSS to ERT in the urine. So then they have developed a score so that we can actually predict the high risk nature of the prostate so that you can avoid repeat biopsy type of things. Okay. So here, with the essence of time, I may not be able to go through the details. So here I want to show you that we developed this RNA in situ hybridization probe for the PCA3, where we have shown that so there are tumors with a different level of expression. You don't see always the 100% of the tumor showing the PCA3 non-coding RNA. We have seen kind of cases ranging from 5% to 100% of the tumor expressing the PCA3. But in this example, I'm showing that is almost every tumor area is showing the high-level expression of the PCA3 demonstrated by the non-coding RNA. And we have actually published a paper combining a study where we were able to correlate the tissue-level expression of the PCA3 with the urine level. Okay, we were able to show the concordance between the urine and tissue level, and we were able to show high concordance, but there are about 5 to 10% of the cases we do found some discordant expression. We were not able to correlate strictly, like when the tissue level may be high, but you may not see enough in the urine, or vice versa, more in the urine but not in the tissues. So we have to learn more about how the RNA is processed from the tissues and coming to the urine. So these are some of the things. But in the majority of the cases, we are able to correlate the tissue level expression and the urine level expression of the PCA3. So please read this paper for more details. Next, we are coming close to the presentation. Here I want to show you that the RNA in situ hybridization method that I have actually shown is not only limited for prostate cancer, and recently we put out a publication showing that the ETV1, ETV4, and ETV5 RNA in situ hybridization can be used in the diagnosis of the CIC, that's type of sarcomas, where you will see the high-level expression of all these three markers combining. So all these three markers, as I showed you before, we don't have good antibodies, only RNA is the best method, and this can be used for the clinical diagnosis of this rare type of sarcomas. And it is not only like protein coding genes or non-coding RNAs. Even some of the Merkel cell carcinoma virus can also be detected by using the RNA in situ hybridization. This is the paper that we published recently. So these are all the application of RNA. As I told you before, this is not limited to one particular application that is for prostate, but depending on the biological question that any investigator you may have, you can actually easily adapt this RNA in situ hybridization method in your lab based on some of the issues that I have discussed. Okay. So in summary, I would like to show that RNA in situ hybridization is a novel approach for the detection of H gene fusions in prostate cancer. ERG-SPINK1, a dual IHC, it revealed a new subtype of prostate cancer with the ERG and SPINK1 expression in the same or independent tumor foci. And by combining dual IHC and the dual RNA-ish, identified new subset of prostate cancer with the dual or triple H gene rearrangements. Then analysis of the whole mount prostate samples revealed the presence of even smaller tumor foci with the heterogeneous molecular markers. And the distinct biology of the H gene-associated prostate cancer suggests that each disease class may require new therapies. Underlying programs controlled by the different molecules. The combined IHC and RNA is a novel approach to screen molecular ablation prostate cancer. And it is important that the combining, so I didn't, since of time, I did not include the slide, but anyone who is interested, please email me that we can do both the DNA fish and RNA-ish on the same slide. If anyone is interested, please contact me. I'll be able to share with you more information about this. I didn't have time to include that particular slide, but we have successfully performed this DNA fish and RNA-ish on the same slide. Okay. So with that, I would like to conclude my presentation by acknowledging the people who are involved in this study. So all the information about the IHC and RNA-ish, we have developed this method in my laboratory, and all the examples that I have shown are actually from the work that is performed in our laboratory. And all the IHC and the RNA-ish work was done by the highly talented staff in my group, Ms. Shannon Koskinen, who is very extremely talented to adopt these kind of new procedures, and we demonstrated it's consistently working well. And I want to thank the pathologists who are involved in the study, Dr. Nilesh Gupta, Sean Williamson, and Dan Chidal, who are extremely helpful by providing the valuable tissue material for our studies. And I want to kind of personally thank Professor Dr. Manimenon, who is the chair of the Department of Urology, who has pioneered in the robotic prostatectomy, the first surgical removal of the prostate by using the Da Vinci robotic system, and who has been extremely, kind of helpful, by providing the excellent support and help by providing the clinical information and the surgical specimen of all the cases that is involved in the study. And our work is supported by funding from NIH, DOD, and Extra Foundation, and the startup fund from the Henry Ford. So with that, I'm thankful to Dr. Chinayan from the Michigan Center for Translation Pathology from the University of Michigan, where I still work as an adjunct assistant professor, and he was actually extremely helpful for developing this type of RNA-ish and during the early stages of its development. So I want to thank everyone. So with that, I would like to conclude my presentations, and I'll be happy to answer any questions. Thank you.
Thank you, Dr. Palanisamy, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how many RNA targets can you detect with dual ISH-IHC?
So as I have shown you in the presentation, right now we have developed the methods for routine use by manual method. We can detect up to 4 molecular targets, like 2 targets by IHC and 2 targets by RNA in situ hybridizations. So we have perfected the method for the detection of 4 molecules in a single experiment. All the 4 molecules can be seen by the bright field application. So if you add fluorescence, we will be able to add more markers. But if I think if anyone wants to kind of restrict their analysis only for bright field applications, right now I think the limitation is for 4. Yes, we can go for additional at least for another 1 or 2, but right now without any issues we are able to do 4 markers under bright field.
Thank you. Our next question is, can dual ISH-IHC be performed on an automated machine?
Yes, as I have mentioned in my presentation, so we have optimized the protocol for the Ventana Discovery XT, which is a research instrument. Even advanced cell diagnostics, they are actually supporting the reagents and the protocol for using the automated Ventana systems. And I think they have another instrument like, I don't remember, it could be a DACO or Leica. The other system can also be used for the automated RNA-ish and IHC.
Thank you. Our next question is, can I do the IHC assay first and then the RNA-scope ISH assay?
No, strictly no, because if you do the IHC first, because of the high temperature and other antigen retrieval procedures, you may kind of spoil the integrity of the RNA by doing the antigen retrieval for IHC. So I would strictly recommend do RNA in situ hybridization first, then do the IHC.
Thank you. Our next question is, can I combine the base scope assay with IHC?
I think so. I have not done this experiment in my laboratory, but I think This question can be best answered by the technical support from the advanced cell diagnostics, but based on the limited information that I gather, I think it is possible to do IHC and base scope. Yes.
Our next question is, are there any recommendations for performing a dual ISH-IHC assay?
So, as I have discussed in my presentations, it is important to know the targets of the target or the location of your targets, like whether the markers that are used for IHC, whether they are going to be like a membrane or nuclear or cytoplasmic, like that. It is important to have the prior knowledge of the location of the target molecules and also the level of expression. Make sure that if you are looking for a marker that is expressed at especially when you are combining like 3 or 4 markers, make sure that they are all kind of expressed at a decent level. If you are looking for a kind of very low expression target, sometimes it may be challenging to interpret the results. It is important that make sure you have some prior knowledge about individual markers when you are doing the combined IHC and RNAs, the locations, and also it is important that, as I highlighted in my presentation, you want to test individual markers first. Make sure that each antibody or the RNA probes are performing well individually in a separate experiments. Don't combine with IHC or RNAs. If it is RNase probe, just do RNAs alone. Make sure that the probe is working and giving the expected results before combining with IHC. So I think I would recommend these are some of the important steps that you may want to follow when you are developing dual IHC and RNase procedures.
Thank you. Our next question is, how many slides can an RNAscope kit run?
I think it depends. I think the amount of reagents and the probe that are provided in the kit, I think one can easily perform up to like a 25 to 30 slides, but it can be stretched depending upon the size of the tissue that you are using. Sometimes you may have a large size tumor or a large tissue size, so you may have to use a little more reagents. If all of them are like needle biopsy, you may be able to stretch up to like 40 or 50 slides. So I think the minimum, I would say 25, maximum you can go up to 40 depending on the size of the tissue.
Thank you. Our next question is, can I use my own situ hybridization probes?
Yes, you can develop your own in situ hybridization probes, but you will be using a entirely different protocol. If you want to use the advanced cell diagnostics method, because you have to use their proprietary probe development system for developing the probes, because they were able to develop the antisense oligo probes, and they develop up to like 10 to 20 Z pairs where they provide more specific hybridization, avoiding non-specific signals, and the Z pairs that they provide, they are specific, and each Z probe has to hybridize in pairs. Only when they hybridize in pair, you will be able to amplify those targets in the subsequent amplification steps. Like that will increase more specificity and the sensitivity for this. But the probes, if you develop your own RNA probe by doing the antisense kind of reverse transcriptions, yes, you can develop it, but I don't think using the ACD bio protocol or the reagents, you may not be able to do the experiment for your probes. So then you have to go with entirely a different vendor, like Promega or some other companies. They provide the RNA detection reagents for this type of probes. So one has to be careful when you are actually trying to use your home brew or the custom-made probes in your lab. You cannot use the ACD bio method. If you want to adapt the ACD bio, you have to have the probe and generated by ACD Bio.
Thank you. Our last question is, how long does the RNA scope assay take to perform?
So normally you'll be able to complete the RNase experiment on the same day. If you start in the morning, by end of the day you'll be able to complete the RNase experiment. This is by manual method. So if you have the automated machines, it takes about, I think, 6 to 7 hours the machine. So when you are using the automated method, you just put the reagents and the slides of the machines, and it takes care of automatically. So it takes about 6 to 7 hours. But if you do use the manual procedures, you will be able to complete on the same day by starting like early in the morning around 8 or 9 o'clock. By 4 o'clock, you will be able to have the slides. So, but in between, when you do the manual procedures, it is heavily hands-on. But of course, you will have in between so many incubations within an hour or 30 minutes or 45 minutes like that. But it is heavily hands-on, but you will be able to complete the procedure on the same day.
I would like to once again thank Dr. Nallasivam Palanisamy. Do you have any final comments?
I want to thank all the participants who listened to this presentation. So please feel free to contact me. I think my email address is provided in the first slide. So please email me for any specific technical questions. We will be happy to share the protocol or send me an email. If you have any specific questions, I'll be happy to answer any questions. If I'm not able to answer, I will direct your questions to the technical service department of the Advanced Cell Diagnostics, and you will get the necessary help. Thank you very much for your time and listening to the presentations.
Thank you, Dr. Nallasivam Palanisamy, for your presentation. I would also like to thank Advanced Cell Diagnostics for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.